Welcome to the Woodside Academy presentation of all things DNA. DNA is one of the most recognizable molecules on Earth. Most people, when they see the double helix of DNA, can recognize it right away, but what they don't know is what goes into actually making the molecule what it is. Usually we find DNA inside a nucleus. About the only time that that's not going to be true is when a cell is dividing because the nucleus falls apart. DNA is made of a double-sided molecule where we have different parts or monomers of the molecule. We call them nucleotides. And, we have to attach them with phosphates. So here I had an adenine and I slid a phosphate over to make a little connection. Notice that it wouldn't let me connect the two unless I had the phosphate. What we have here is a sugar and a phosphate, and that makes our backbone for our DNA. So every time I'm going to attach a new nucleotide, I have to put a phosphate in it first. And I just go along. And this is making one side, or one half, of the DNA double helix. Now on the other side, we're going to get to match up the double helix with the proper nucleotide that will join with the nucleotides that I have here. So notice I have A, C, C, G, G, and I'm going to have T, T. That stands for adenine, adenine, cytosine, cytosine, guanine, guanine, and thymine, and thymine. And there we go. See, we've actually made an entire DNA molecule, one side of it. But how about that other side? Well, notice that now I have things to choose from over here in my nucleotide pool. So you'll notice that I can't possibly bind something like this cytosine to that adenine. It just won't fit. But notice that this thymine will come over and fit very nicely, because adenine always binds to thymine. So look, I have another adenine. That means I'll need another thymine. Can you tell what's going to bind with the cytosine just by looking? Of course you can. It's a guanine. And so we just go down and we make the two sides of the DNA double helix by matching up adenine to guanine, adenine to thymine, cytosine to guanine, and vice versa, guanine to cytosine, and thymine to adenine. Interestingly, the bonds in the middle here are actually hydrogen bonds, but the ones that are up the sides with the sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, those are covalent bonds. And you should remember bonding. We talked about that when we talked about general molecules and water. If not, you might want to go and review that video clip in the Woodside Academy. So there we go, and we have made the DNA double helix. Now when cells divide, they actually have to replicate this and make two copies of it. And they use a special enzyme called DNA helicase. Enzymes always end in ASE or ACE for enzyme. And it's a DNA helicase because the enzyme is going to work on the DNA helix and unwind it. So if I just release this enzyme, goes over, look, oh, it just undid all my work. Ugh, blasted enzyme. So now we're going to make new DNA using another enzyme called DNA polymerase. We're going to make a DNA polymer, so we're going to use DNA polymerase. We got to click and drag everything. Okay, so here we go, and we're going to click and drag our nucleotides over here to make new strands of DNA. Notice that each of our new strands is going to have part or half of the old strands. We actually call that semi-conservative replication because we're conserving part of the old molecule. So see how I'm dragging over and I'm making a whole new molecule, and it's going to look exactly the same as the molecule I started with when I'm all done, except half of it's going to be old molecules and half of it's going to be new molecules. And this is what goes on in cells when they're replicating themselves and doing mitosis. You have to make a copy of this DNA so that every single cell gets a copy of the DNA. You have 46 chromosomes full of DNA, full of genes that are coding for all your traits. And every time they divide, they have to do this little trick where all the DNA gets replicated.